Thank you all to continue with us. I am Teresa Rivas, as you may know, a PhD student from BrainLab. And in this section, I have the pleasure to present you 4 female speakers from different international universities and scientific career stages. All of them will briefly present us their research line, and at the last part of the symposium, they will participate in a roundtable discussion. Before the presentation of the first speaker, I would like to remind you that questions can be written in the question chat and will be addressed to the speaker at the end of each talk.
Sorry, we hear a lot of echo.
Maybe like this?
Do you still hear you better?
Thank you. I'm sorry again.
But it will still like a lot of echo.
Sorry?
There's a lot of echo. It looks like you're in a church or something.
Okay. Now? Is better?
No. Axel, can you?
Yeah, I can hear you. Should I start?
No, Axel doesn't have an echo, right?
No, well, I hear you both perfectly well.
I don't know, maybe Axel doesn't have an echo, but I have a horrible echo.
Should I?
I'm sorry, there are a lot of technical problems.
No worries.
Now it's better?
Now?
And Dere, can you try it?
Wait, maybe it comes from me actually. That's good. And is that better?
Can you hear me now?
Yeah, I can hear everyone. I can hear everyone fine actually.
We can hear you fine, so I think that we can, if Dere can give a short presentation, we can go on and we will try to figure what's happening.
Yes. Okay. The first speaker will be Axel Karkus. Axel Karkus obtained her PhD in 2015 at University Libre of Brussels, and after that, he worked as a postdoctoral researcher for 5 years. Axel has recently returned home as an assistant professor of the University Libre of Brussels, focusing on auditory cognitive neuroscience. In the present symposium, she is going to present her research on the developmental effects of hearing loss on speech processing. Once again, Axel, thank you so much to be here.
No, thank you. It's really a pleasure to be here, and I'm really honored and really happy to take part in this day. Today I'm going to present some of the work I did actually 2 postdocs ago. So just don't worry if you're really slow at getting your data published, because we all are. So this is work I've done at UCL with Lorna Halliday and Stuart Rosen, focusing on the neural perception and processing of speech in children with mild to moderate sensory neural hearing loss. And today, for the sake of time, I'm going to focus on the processing of speech in this talk. And so when it comes to severe to profound hearing loss, we know that the structure and function of the auditory system depends upon stimulation that's received during maturation. And we know that from animal studies that have shown changes at various levels of the auditory pathway, from the cochlear nucleus to the auditory cortex, following severe to profound deprivation. But we also know that from studies conducted in children with conductive hearing loss, which you typically have when you suffer from otitis media, for example. And we know that conductive hearing loss leads to temporary changes in at different levels of the auditory pathway in children. Why are we interested in mild to moderate sensorineural hearing loss? It's because it has a really high prevalence, actually. It's 1.46 over 1,000 live births. And even if those children only have mild or moderate sensorineural hearing loss, so that means thresholds from 21 to 70 dB in the audiograms, they are at higher risks. So, I mean, they do have residual hearing, right? But they are at higher risk of experiencing language difficulties and general auditory processing difficulties. And so the starting point of this project was to try and figure out how does a peripheral impairment affect central processing of sounds in children with mild to moderate hearing impairment. A good paradigm to study this, this question is called the Oddball Paradigm. And so that's neurophysiological or EEG recordings in response to a stream of sounds that's represented here with the letter S for standard, and then the letter D represents the deviant. And so in practice, it would sound like this. So I hope the sound is going to work. You let me know. So I don't know if you heard that, but the standard was the syllable ba, and the deviant was the syllable da. And in this case, there were actually two deviants that are slightly different one from the other. There's one that's very obvious, because it's at the other end of the voicing continuum. But then there's another one that's less obvious, and it's kind of in between a ba and a da. What's interesting is that when we look at the typical response, electrophysiological response evoked by standards and deviants in young children, in adolescents, this is what the response to the standard looks like in one 14-year-old normally hearing child. So I don't know if you're familiar with these responses, but typically, so you see a vertical line at 0, that's the beginning of the sound. And then we've averaged together 600 repetitions of the standard. And what we see is that there are 4 very typical replicable peaks that are shown here, P1 and 1, P2 and N2, and those are the cortical exogenous, we call them sometimes obligatory responses. Interestingly, when you present and when you look at the response to the deviance, this is what you get. So the black line is always the response to the standard, and this gray line here shows the response to the deviant, and we can see that it starts to be quite different from the standard, from around 150 milliseconds onwards to about 400. And so that portion of a difference and a larger negative response evoked by the deviant is called the mismatch negativity. Interestingly, so this is what we see in typical adolescents, but in younger children, the morphology of the response is different. So this is what you would see in an 8-year-old normally hearing child. And you first have this positive peak, and then directly after that, you have a large negative deflection. And those are going to become the P1 and N2, but they're not quite there yet. It's kind of like this is the typical response in younger children. And you might notice that there's no N1, no P2. Those two responses are going to grow later. And this is due to the maturation of the generators of all those different peaks. So using this paradigm, we investigated quite a large cohort of children. So when I say large cohort, you might see here in the bottom right section of the table that we tested 90 children. More or less half of them had a mild to moderate hearing impairment. And I'm going to simplify and say hearing loss now, or hearing impairment, but I really mean mild to moderate. So those were 46. And then children with normal hearing, so NH, they were 44. And because this was still quite a large cohort and we were interested in the developmental effect of hearing loss on the neural responses to speech, we actually split those two groups between older children that were 7 to 11 years old, younger children, and older children that were 12 to 16 years old. And so very briefly, they were all monolingual British English native with nonverbal IQ over 85. It was a 2-deviant oddball paradigm that I just shown you. The sounds were presented at 70 dB SPL, and children with hearing loss were tested without their hearing aids. So obviously, with mild to moderate hearing losses, you have thresholds up to 70 dB HL, but then presenting the sounds at 70 dB SPL in the worst case scenario. So for children with the poorest thresholds, they would still be close or above their threshold. And it was quite a large testing. Actually, children came twice to the lab. And a portion of the testing was strictly behavioral discrimination tasks that I'm not going to talk about, at least in my presentation, but we can talk about it in the question time. This is just to show you the difference in audiograms, but in short, children with hearing loss, the younger children and the older children had comparable thresholds that you can see here in orange. And this is a good rule of thumb, not rule of thumb, but this is a good habit you can get for this presentation. Orange is always children with hearing loss, and blue is always normally hearing children. What do we see in terms of the results? Well, I know this is a bit dense, so bear with me. We first look at the upper row, and those are the normally hearing children. The black line is always the response to the standard. The dark gray plain line is the response to the large deviants, and the dotted line we're going to ignore. That's the response to the small deviant that's very tenuous, let's say. And the results for the small deviant weren't that obvious. It didn't really elicit a clear MMN, so we just didn't include it in the analysis in the end. But what we see for young normal hearing children, so 7 to 11 years old, in response to the BADA oddball paradigm, is that we have the P1, we have the N2, but most importantly, we do have a significant MMN for a portion of time that's indicated with the segment of line from around 250 milliseconds to a little bit after 300 milliseconds. So everything's fine and in place there. In the older children with normal hearing, again, we see a present MMN for a portion of time from around 200 to 300 milliseconds, so perfectly right. And now in the lower panel, on the left side, you have the younger children with hearing impairments, and we do see an MMN. It starts a little bit, well, it's actually very comparable here in the speech condition. So we do have an MMN, and it's comparable to age-matched normally hearing children. The interesting part of this plot is the lower right panel. That's the responses in children with hearing impairments, but older children with hearing impairments. And as you can see, there is no significant MMN there. Interestingly, actually, this project had started before I joined in. So it started in 2011, and I joined the lab in 2016. And so by the time I got all set up, we were actually able to call a portion of the young children with hearing impairment that were initially included in this study in 2011, but we called them back 5 or 6 years later. Sorry, we're gonna ignore that now. And so this is what you can see here in the upper row. So on the left, it's the subset of children who were initially tested in 2011 and able to come back in 2016. And as you can see, the shape is a little bit different because we only had 13 or 14 children who came back from the initial 24 children sample. But then of those children, we tested them again in 2016, and that's what you can see on the right upper row of this plot with the blue labels, and that's the follow-up. And as you can see, there is still an MMN, but it's a lot later, it's smaller, and it definitely didn't mature like the response did mature for normally hearing children. And that's what you can see in the lower left panel. So those are the older children with normal hearing. And then just to refresh your memory, on the bottom right, you have the initial sample of 24, 23, I don't remember, children with a hearing impairment. So clearly we have a big difference and an evolution and a detrimental effect of hearing loss underrepresentation of the MMN evoked by speech material in children with hearing impairment. But at this point, you could say, well, okay, but you tested the children without their hearing aids, without any kind of help, really, and that's not really representative of their everyday life. So in a second study, we investigated how amplification may help and may benefit the neural encoding of speech in children with hearing impairment. So we've talked about cortical processing so far, and we've added subcortical processing in this second study based on the FFR. So I know we've talked about this before, so I'm not going to go into too much detail, but just to remind you, the frequency following response is evoked by sounds at the level of the inferior colliculus, and it's a faithful representation of the fundamental frequency and the first harmonics of complex sounds such as speech. So what we did here is that we tested a much smaller cohort, unfortunately, so about 40 children, half of them with a hearing impairment and then half of them with normal hearing. And again, you can see the audiograms here. We used the 1D Oddball Paradigm, presented the sound at 70 dB SPL. Children with hearing impairment were tested without their hearing aids, but they were presented with two conditions, and that's what you're going to see here. But first, let me show you the results for the controls. So in the upper panel, you see the response to the envelope, so the lower harmonics, and the fundamental frequency of the bar syllable was at 220 Hertz. And that's exactly where you see the first peak in the response here. And then you have the first harmonic at 440. I'm not going to focus on the TFS at this moment. I want to move on to the responses and the results in the children with hearing impairment. So first, children with hearing impairment presented with the sounds at 70 dB SPL with no amplification whatsoever. Obviously, they're impaired. The rest, the FFR is smaller at both the F0 and H2. And at this point, it's really just a smaller response than the controls. Interestingly, we presented children with hearing impairments with two different conditions in this study. The first one, stimuli were unamplified, and the second one, stimuli were amplified. So we simulated the gain provided by their own hearing aids based on their individual audiogram. And in that case, what we see is that we do have benefit of amplification, definitely at the F0 that's now comparable to the F0 of controls, but also H2, and we see a large benefit in the really high harmonics in the TFS. I'm not going to talk too much about this right now, but again, we can discuss that in the question section. So just a quick note, still in line with this idea of a developmental effect of hearing loss on the neural processing of speech. Here we can see the relationship between age and the amplitude of the FFR at F0 and H2. Interestingly, I want you to look at the lower left panel, which is the relationship that we see with the amplified stimuli for the children with hearing impairment at F0. And we see that there seems to be a bigger benefit for younger children than older children. So again, there seems to be a developmental effect of hearing loss, this time on the benefit provided by hearing aids. Now, if we look at the at the cortical responses, you're used to looking at these now. The light trace represents the response to the standard, and the darker trace represents the response to the deviant. And so in controls, you can see again, there's this dark line, black line from around 350 milliseconds to 440. And that represents a significantly present MMN. As you can see, we didn't observe a significant MMN in the children with hearing impairments, neither when presented with unamplified stimuli nor amplified stimuli. So there was no significant MMN in this group now. So if I summarize all of the results that I've shown you so far and that I've shown you today, we can say that even just a mild to moderate degradation of the auditory input affects the neural processing of speech at both subcortical and cortical levels. Interestingly, when we provide hearing aid amplification, it significantly improves the neural processing of sounds at the subcortical level for sure, but not at the cortical level. And that observation is in line with previous studies that had been conducted in adults and in animal populations. The way I interpret this is that actually there's increasing multisensory integration at successive levels of the auditory system. So up until the inferior colliculus, the auditory pathway is strictly unimodal. But then after that, it becomes multimodal and integrates information from other sensory modalities. And I believe this is the reason why there is a benefit at the subcortical level, but not so much at the cortical level, probably because there has been takeover in a way from another modality. So merci beaucoup. And let me know if you do have questions.
Thank you so much, Axel. And now it's turn for questions. The first one
is from Night Making, and say, thank you for this interesting study. Could the missing MMN in the second study be due to the mixed age group?
Yeah, for sure. The problem is because the sample was a lot smaller, we couldn't do the split between the younger and the older. But when I look at the relationship between amplitude and age, amplitude of the MMN and age, I can see the same trends toward the developmental effect of hearing impairment on the amplitude of the MMN. So as children grow older, their hearing loss somehow affects the representation of the sounds and the discrimination of the sounds.
Thank you so much. And there's another one. Well, there are several ones, but maybe the next more what is, could the children differentiate the stimuli you used in the MMN study?
Yeah, that's a really good question. So as I said, we had behavioral tests as well. And in the behavioral tasks, children with hearing impairment were poorer than controls, as expected, but we didn't see this age effect. So their performance didn't drop as they grew older. So that's really puzzling, obviously. And we think it's probably due to compensation somehow, that obviously the MMN is a good index of neural discrimination, but it's not, there's not a linear relationship between the amplitude of the MMN and the behavioral performance. So we really believe that somehow something else comes into play that potentially helps the children actually perform the task. So at the behavioral level, they're still, well, they're not good, but they don't get worse as they grow older, which is good news for them.
Okay, thank you so much, Axel. And there are a lot of questions, but maybe now we don't have a lot of time. If you want it, you can answer in the chat.
Sure.
And we can continue with the next speaker.
I will do that. Thank you very much. Thanks for having me.
Thank you so much, Axel. Thank you again. Okay, the second speaker will be Sophie Hurts. Sophie obtained her PhD in 2014 at the Berlin School of Mind and Brain. Throughout the years, she worked as a postdoctoral researcher at the Max Planck Research Group Auditory Cognition, at Research Group Auditory Cognition, and later at the Cognition and Brain Dynamics Group. Since 2019, she has been working as a junior PI in the Cognition and Brain Dynamics team. Her research focuses on implicit and explicit timing processes, and specifically how implicit temporal prediction is formed from temporal statistics of auditory input. Sophie, thank you for having accepted our invitation.
Thank you. Can you hear me?
Yeah, for sure.
Okay. Try to share my screen. Okay. Can you see the slides?
Yes, perfectly.
They also move?
Yes, they
okay,
move.
great. Well, I'll just start.
Okay,
Thank
whatever
you.
you want. All yours.
So I would like to thank the organizers, first of all, for putting together this amazing event. I really think the previous talks were all super interesting and very nice to follow. So I'm having fun. Thank you. I would like to draw your attention to time for the next 15 minutes. So in the Cognition and Brain Dynamics group at Neurospin, headed by Virginie van Wassenhoff, we are interested in temporal cognition in different aspects. So how does the brain represent and use time? So first of all, what is time? Well, I cannot answer that question, and a lot of philosophers and physicists and other people have tried, and we don't have a clear answer for now. What we know is that there's no material substrate for time. It's not like something we can measure in the environment. So we have developed, humankind has developed all kinds of clocks, so time measure devices. I've put some examples for you here. But the thing is, they don't really measure time. Rather, they create time. So they have some inherent metric that creates regular time intervals or ticks. And by counting them, we can have a universal measurement of time. But yeah, they don't extract time from the environment. And so the question is, how does the brain do it? So before we look at the brain, let's have a look at what is time for the brain and for the mind. Why do we need time? So time can be represented as order. For example, we can say one event happened before another, or by synchrony. So we can synchronize our movements to external inputs, or judge whether two events happened at the same time or not. And importantly, and that's going to be the focus of my talk today, there's duration. So duration is the empty space between two events. I'll try to play that. I'm not sure if you can hear that. So what you should have heard are just beeps, like in the little cartoon. So you heard beep, beep, and then another one, beep, beep, and then you would have no problem telling me that the first duration was shorter than the second, even though the sensory events, the tones were the same. So what I want to say with this is that our brains can create a percept of an empty time interval, and we can compare these, and yeah. So the question that has motivated my research since my PhD is, when we ask humans to estimate time, we're often quite variable, and our estimates don't match with clock time. So this is on the right-hand side, you see a plot from my PhD, where I played visual flicker to participants. So I stimulated their eyes with different frequencies. And what we found was that especially the lower frequencies caused a big dilation when we asked them to estimate the duration of the stimuli. So yeah, whatever you do during a time interval has a big effect on the subjective perception of this duration. So with respect to clock time, it's not very valid. But then on the other hand, humans can efficiently extract temporal regularities from sensory environments, for example, from sound. So when we listen to music and the note is slightly delayed, we will easily notice that. And that is something that has been studied under the umbrella of implicit timing. And here on the right-hand side, I show you a paradigm that I used during my postdoc, where I played pairs of tones to people and asked them to judge the pitch of the second stimulus. So was the second tone higher or lower than the first? And the interval between the two could either be predictive or non-predictive. And importantly, what we found in these studies is that when the time interval is predictive, so we can predict only the onset of the second tone, not its pitch, the pitch discrimination becomes more sensitive. So you see that the slope of the green curve is steeper than the slope of the blue curve. And this is without telling participants anything about the timing. So yeah, we can very efficiently extract these sort of fine-grained temporal regularities from sensory environments and use them. So why are these two timing processes so different, or are they? And to summarize, so what I just said, implicit timing is the extraction of temporal regularities for timing tasks where time is not the main task goal, so you're performing a sensory task. And you form temporal predictions about durations. And on the other hand, explicit timing is when we have to make an over judgment of time. So when we ask to say how long an interval lasted. And the parsimonious assumption would be that the brain uses one representation of time or duration for these different situations. But the existing literature shows that there are quite some divergences between the two processes. So just to give you a few examples, some studies have shown that the variability of implicit and explicit timing processes overlap, as you see in the picture on the right-hand side. But then others have refuted that. And importantly, we seem to see across the literature that implicit and explicit timing doesn't really correlate across participants. A very seminal fMRI study has shown that there are different brain areas involved. So when participants perform a production task, that means they have to explicitly produce a time interval versus when they have to predict implicitly. We see for the production task, we see motor areas, the pre-supplementary motor area and prefrontal cortex, while the prediction task seems to be more represented in the parietal cortex and the cerebellum. So apparently there are different brain areas involved. So the fMRI study here, we don't have a temporal resolution that can tell us at which processing stage these two processes diverge. So when we look into the EEG and MEG literature, there are studies either on implicit or explicit timing, which show that, for example, the beta band plays an important role in both processes, but there's so far no study that compares the two timing tasks in one. So that is what we did here in this study. The paradigm is very similar to what I showed for the implicit timing study. So participants are presented with tone pairs, and they have to say whether the second tone is higher or lower than the first. That's the implicit timing task. And we again vary the foreperiod, that is the interval between the two tones. So it can either be the way that the second tone is predictable in time, or it's not. And then we add top of this an explicit timing task, where instead of asking participants to judge the pitch, we ask them to judge the duration, to say if the interval between the two tones was long or short compared to all the intervals that they already heard. And this is in the same session, but in two different blocks. So here you see again the schematic representation. So it's a 2x2 design. We vary, on the one hand, the temporal statistics to be either temporally non-predictive or predictive, or we vary the task to be either pitch discrimination or duration discrimination. And so this gives us these 4 conditions where here you can neither time implicitly nor explicitly. Here, when the stimulation becomes predictive, you can use implicit timing. Here, you're supposed to use explicit timing, and then in this last condition, both implicit and explicit timing should be at play. So behaviorally, what we find is that, similar to the previous studies, temporal predictability speeds up response times, both in the pitch discrimination and in the duration discrimination task. So you can see here that the little dots are a bit below the unity line, so response times are longer in the non-predictive conditions in both tasks. And you can see here that that's modulated by the 4 periods. So the biggest advantage happens at the earliest 4 periods, which is very common in the literature because that's when you're most surprised. So when your prediction is most helpful. Now we looked at the sensory discrimination performance, and we replicate the effect of the previous study, namely the increase in pitch discrimination sensitivity. So tones that can be predicted in time are judged more accurately than tones that cannot be predicted in time. And a bit to our surprise, we didn't find an advantage for the duration discrimination task. So no matter whether the interval was to be predicted or not, participants performed the same way when they had to judge the duration. Oops, sorry. Yep, I hope you can still see it. Now we're going to go to the MEG recording. It was performed while the participants were in this study.
We can see perfectly.
Okay, sorry. Yeah, it's just a bit weird to hear no one, but it's fine. Just let me know if you can see it. So, okay, I wanted to talk about the MEG. So we record the brain activity. So MEG is very, in terms of what it records, is quite similar to EEG, only that it records the magnetic field and not the electric activity. So we can have a very fine-grained temporal resolution of the processes. So first we looked at how participants' brains process the Q tones, which either gave a temporal prediction or not. And we find that, especially in auditory areas, which you see here in the topography and here in the source plot, that there is a distinction for predictability. So cues that predict when the next target will come have evoke higher auditory activity. So that's the dark red and dark blue lines, compared to cues that don't convey a temporal prediction. And that's particularly strong at the short 4 periods, which is nicely in line with the behavioral data. Now we're looking at the target. So a tone that was predicted in time versus not. And again, we find a distinction in auditory areas, namely that tones that could be predicted result in higher auditory activity compared to tones that could not be predicted, regardless of the task. So, yeah, in line with the response time data, these evoked potentials, evoked fields in this case, show that the temporal prediction was processed no matter the task. So both in the pitch and duration discrimination task, temporal predictions were used. But yeah, as I said before, apparently it only helped the discrimination of the tones and not the discrimination of the intervals. There was an effect of tasks, but it happened very late. So yeah, like about 500 milliseconds after the target, the activity in auditory areas was still higher than for the pitch discrimination task and for the duration discrimination task, which could reflect some prolonged processing of the auditory input if the task is to judge the pitch compared to judging the duration. Now I'm moving to oscillatory signatures. And importantly, now we look in the interval between the cue and the target. So from the results before, we've seen that the temporal prediction is processed, but we don't really know how it's processed. So how is it maintained throughout the 4-period interval? That's the question. And here we find 2 effects. This slide is a bit crowded, but I'll try to walk you through. So again, we're looking at the target and the time window before that. And what you see here, the lines reflect the time courses of beta band power. So beta is usually between 15 and 25 hertz. And what you see is that the beta power is higher for the pitch discrimination task, so for the red and orange lines compared to the duration discrimination task. And you also see that it's higher for the predictive conditions in both tasks. And this happens over motor and parietal areas. So this is a signature of a task difference in the study. And then we find also an effect of predictability, which is reflected by alpha power. So it's higher, alpha power is higher prior to the target for predictive conditions compared to non-predictive conditions over, again, parietal, but this time also reaching into visual areas. And that's a common finding in the timing literature, that we have these preparatory alpha effects. And since it's an auditory task, the direction is this way. In a visual task, you would have expected to see a decrease, but in an auditory task, we usually see an increase prior to a temporally predicted stimulus. So I've lost track of time, but here at the conclusions now we can go more into the details if you want. So in a nutshell, this study shows both diverging and converging effects for implicit and explicit timing. So implicit timing, again, seems to be processed in a very automatic manner. So it speeds up responses in both tasks, but it only improves pitch discrimination. Then implicit and explicit timing show early versus late effects on the processing of auditory stimuli. Parietal alpha power index temporal predictability during the 4-period, and sensory-motor beta activity reflected the task difference. But surprisingly, we found no positive difference in the neurosignatures for explicit versus implicit timing. So what I mean is there was nothing that we found was higher in the explicit timing task during the 4-period. So this could be a first hint that at least during the 4-period, when we are either implicitly or explicitly timing, there is a shared representation of time. But of course, further research needs to be conducted. And in this case, especially the fact that both tasks were performed within the same session might have led to some overlaps here. So, with this, I'd like to thank my collaborators, the Brain Dynamics and Cognition Group, Virginie van Wassenhoven, and my former postdoc advisor, Jonas Oblizer in Lübeck. Thank you.
Many thanks, Sophie, for your brilliant presentation. The audience are so interested. And one of the questions is from Stephanie. I love your talk. I was wondering if you can make these implicit-explicit timing comparisons also in animals.
That's a good question. I don't know of any study that did the comparison, but I for sure know that there's implicit and explicit timing studies in animals. So actually the timing literature has a lot of animal work in the 60s, 70s. And for example, pigeons or rats are very good in timing an interval to get a reward. So they can have to press the lever. When they press exactly after 10 seconds, they'll get a reward, and then they press too early or too late, they don't get one. So I think the comparison of paradigms would definitely be feasible. Now it's of course a big question what the explicit timing, in a way, it involves consciousness, and to what extent does the animal consciously represent the time interval. That will be more difficult to know, but yeah, I think it definitely is something that could be done.
Okay, thank you, Sophie. The next one is, do you think that time perception in implicit or explicit talks can be affected by the language the participants speak, or is it a dominant general ability?
I would say that, especially the implicit timing, I would think it's strongly domain general. I know that there are some studies by Lera Boroditsky who looked but more at the explicit representation of time, and they looked at cultures where, so we, in our Western culture, we have this strong habit to represent time from left to right. And so it's interesting to look at cultures where, for example, they don't write from left to right, or there's some cultures that don't represent space in absolute coordinates, but always in representation to themselves. So when you, for example, you ask people from these different cultures to align pictures, someone from Western culture would align them from left to right in time. So say you have a picture from a baby to a grandfather, you would align them in a different direction than someone from a culture where space is represented differently. But that's I don't know of any cultural differences in any more implicit timing tasks.
Okay, thank you. And the last one, what were the pitch frequencies used?
So the frequency used were between 500 and 900, I think, hertz. And it was pure tones, but we titrated individually to make the task at a performance of 75%, more or less. So every participant underwent a little training procedure, and those who were very good in discriminating the pitch, we reduced the differences. So the actual pitch differences were quite small, like a hertz or something for some participants.
Okay, that's it. Thank you so much, Sophie.
Thank you.
We will see you again in the roundtable.
Yes.
Thank you.
Bye-bye. Thank you.
Bye. Okay. The third guest will be Konstantina Kilteni. Konstantina studied electrical and computer engineering in the National Technical University of Athens, and she did her PhD in health and clinical psychology using virtual reality in the Event Lab at the University of Barcelona. Konstantina was a postdoc in the group from 2015 to 2019. After that, she was promoted to assistant professor, and a few months later, she set up her independent group at the Department of Neuroscience from the Karolinska Institute. She is currently awarded with some important grants and prizes, such as the Strat Neuro Startup Grant in Neuroscience or the Swedish Research Council Starting Grant in Medicine and Health. Konstantina Lab focuses on studying how the brain predicts the sensory consequences of our actions. And many thanks, Konstantina, for being with us.
Thank you very much. Can you hear me?
Yes, perfectly.
Okay, thank you very much for the invitation. I'm very happy to be part of this initiative. Today I want to share with you some unpublished findings on somatosensory perception during action. Can you see my screen?
Yes, perfect.
Very nice. And you see this change in the slides, right?
Right.
Okay, great. So I want to let you know what are my research questions. So I decided to put an example, an everyday example. Let's assume that you experience pain on your foot. So you go to your doctor's examination office and you want to tell the doctor where you experience. You want to point where you experience your pain. So you move your hand and you press with your index finger on a specific location on your foot. So you generated a pressure, so it's a self-generated pressure on your foot. Now let's say that the doctor wants to confirm the location, so she moves her hand and she applies pressure with her index finger on the same location of your foot. So in this case, you experienced an externally generated pressure, so it's a pressure that the doctor generated on your foot. And let's assume that these two scenarios were while you were resting comfortably on the examination bed. So you experienced the self-generated or the externally generated pressures on your foot, on your resting foot, Or the doctor asks you to move your foot while you experience these self-generated or externally generated pressures to see if the movement influences the pain, for example. So you can see that there are 4 different scenarios. You can experience a touch that is self-generated or externally generated, and your body part that receives this touch can be either at rest or moving. And I'm interested in how the central nervous system can differentiate between these 4 scenarios. So there is one well-established phenomenon in the motor control domain. It's called the somatosensory attenuation phenomenon, and it concerns self-generated strokes, self-generated forces, or self-generated taps, so self-generated touches. And this phenomenon describes how these self-generated touches feel weaker than externally generated touches of the exact same intensity. Now, this is in the behavioral experiments. Neural experiments have shown that this attenuation is reflected in an attenuated activity in the secondary somatosensory cortex and the cerebellum. And a recent study found that the more you attenuate perceptually your self-generated touches, the stronger is your functional connectivity between the cerebellum and the secondary and primary somatosensory cortex. And this is a phenomenon that it has been found also across the animal kingdom, so from crickets that suppress self-generated sounds to monkeys that suppress self-generated vestibular information. And a recent study in human found that this phenomenon is present in 98% of a big sample with 322 people with a very wide range, age from 18 to 88. Now, there is another phenomenon, again in the motor control domain. It concerns, again, touch. You can find it as a term as movement-related tactile gating or movement-related sensory suppression. And this phenomenon concerns externally generated stimuli that are applied while you move. For example, you might be asked to extend or flex your elbow while you receive electrical stimuli on your wrist. Or you might be asked to abduct your index finger and you receive electrical stimuli during this movement on your index finger. Sorry. And this phenomenon describes the elevation of the detection thresholds of externally generated stimuli during movement, as well as a weaker perception of these externally generated stimuli during movement compared to rest. And this is in the behavioral experiments. Neural experiments that you can find even from the 60s, they have shown that this phenomenon is represented in an inhibition in the amplitude of the somatosensory evoked potentials. So the amplitude during movement is greatly diminished compared to the somatosensory evoked potentials of the same exact stimuli during rest. So these 2 phenomena, these 2 suppression processes, let's call them like that, they share many dissimilarities, but also many similarities. So if we look at their dissimilarities, attenuation is discussed for self-generated stimuli. Gating is discussed for externally generated stimuli. Attenuation concerns both the limb that moves and the limb that receives the touch, while gating concerns only the limb that moves. There are also different movement types that can be studied in attenuation and gating. So attenuation is observed only during active movements, while gating is observed both during active and passive movements. And there are experiments showing that there is a different temporal sensitivity. So there is a very narrow time window within which you need to receive the touch in order to attenuate it, while gating you can observe during, but also even before the movement. With respect to similarities, both phenomena, they have been proposed to modulate tactile perception during movement, and they have been proposed to have the same function. So you need to suppress sensory information incoming because you need to prioritize either external stimuli in case of attenuation, that can be from your predator, so they might be more relevant, or you want to prioritize stimuli that are more task relevant in the gating literature. And both phenomena, they have been shown in various species, and they both have clinical relevance. For example, functional movement disorders, schizophrenia, Parkinson's disease. So it's not surprising that many researchers, they use the terms interchangeably, and some of them consider them to be the same suppression process. However, there is no experimental study that has tried to combine these two sensory processes within the same experiment, using the same stimulus and using the same psychophysics task. And this was the purpose of the study I'm going to present you today. So we designed an experiment that had 4 experimental conditions, and we manipulated 2 factors. One factor was whether you received an externally generated tap on your left index finger, so the motor pressed on your finger, or you received a self-generated touch on your left index finger, so you moved your right hand, you tapped a sensor, and you produce a touch on your left index finger. So first factor was that. And the second factor was whether the limb that receives the touch, so the left arm, was at rest, or the left arm was at movement when it was receiving the touches. So I will go quickly through the names of the condition. So we had a baseline condition in which you received an externally generated tap on a relaxed left limb. The attenuation condition in which you received a self-generated tap on a relaxed left limb. The gating condition in which you were moving your left limb, and during this movement you received an externally generated tap. And the attenuation and gate condition, which combines these two. So you receive a self-generated tap while you are moving your left limb. In all 4 conditions, the participant received 2 taps on their left index finger. The first one, the test tap, is the one that we manipulated, whether it would be self-generated or externally generated, or whether you were moving your left limb or not. But the comparison tap, the second tap, was applied after. It was always applied when you were at rest. So in the 2 conditions in which the participant had to move their arm, we had our equipment on top of a platform that could very easily move forward and backward on top of some commercially available drawer runners. There was a starting position and an end position, and we also put a motion sensor on the platform so we can track the position of the participant hand during time. And here in the middle panel, you can see the position of the participant have in all 70 trials for 1 condition. Now, as I told you before, the participant in all conditions received 2 taps. The first tap had a fixed intensity of 2 newtons, and it was the one that could be self-generated or externally generated, or could be applied while you move or not. And the second tap, the comparison tap, varied its intensity from 1 to 3 newtons on every trial. And for every of the 4 conditions, we fitted the participant responses. So we asked them which you think is stronger, the first or the second. So we collected these responses and we fitted them with logistic function models. And we estimated 2 parameters of interest: the point of subjective equality that corresponds to the intensity of the comparison tap at which you feel that the test tap is equal to, and the just noticeable difference, which is a representation of the slope. So lower values of point of subjective equality mean that you perceive the test tap weaker, while higher values of just noticeable difference represent that you need bigger difference in the stimuli to perceive them differently, so you have a worse performance. You have a lower precision, sensory precision. And we had 24 subjects, so we fitted the participants of all subjects, and we extracted these 2 parameters for every condition and every participant. So with respect to the point of subjective equality, we observed that the self-generated touch, which you can see in the red bar and the yellow bar, felt significantly weaker compared to externally generated touch, both when your arm was still and your arm was moving. The line plots that you see here reflect what I just told you. So the first one here shows that the self-generated tap feels weaker than the same tap when it is externally generated when you are at rest. And the same pattern, a self-generated tap feels weaker than an identical externally generated tap when you move your arm. We searched for correlations between the point of subjective equality, and actually the only significant correlation was between these two conditions, indicating that the weaker you perceive a self-generated touch when you are at rest, the weaker you perceive a self-generated touch when you are moving. So to summarize, we found a decreased magnitude, PSE, of sensory reafference, so self-generated touch compared to externally generated touch. And then we looked at the slopes, so the JNDs, and we found that as soon as the limb that receives the touch moves, your discrimination capacity is worse. And you can see in these two conditions here that involved the movement of the left arm. And again, the line plots here suggest that your discrimination capacity is worse for an externally generated touch when you move compared to when you don't move. And the same applies for a self-generated touch. You discriminate less when the touch is self-generated and you move compared to when the touch is self-generated and you don't move. Again, we found the only significant correlation was between the slopes of the two conditions in which you move your arm, indicating that the more difficult for you to discriminate touches, externally generated touch on a moving limb, the more difficult it is for you to discriminate also self-generated touches on a moving limb. So we found a worse precision, JND, in processing both sensory reafference and sensory exafference during movement compared to rest. Here you see the group psychometric curves for the four conditions. You can appreciate that the two conditions, the first two, the gray and the red, the baseline and the attenuation condition, it's just a shift of the curve to the left, suggesting this a reduced perception of the self-generated touch. But you can see the other two conditions, they have different slopes, they are flatter because it's more difficult for you to discriminate the touches. But again, you see this shift in the curve for the self-generated touch, suggesting that you perceive it weaker. To further illustrate these differences, we try to predict the participants' performance in the condition that involved attenuation and gating using the parameters of the attenuation and the gating condition. So when we use the point of subjective equality of the attenuation and the JND of the gating, we were able to predict better the participant responses that you see here on the dark blue markers than if we're using the other way around, the PSE of the gating and the slope of the attenuation condition. And this comparison was done using the Bayesian information criterion, so which model predicts the data better. So to conclude, I think this data suggests that attenuation and gating are distinct perceptual phenomena. So you have a decrease in the perceived magnitude for self-generated touch, but you have a worse precision during movement for both self-generated and externally generated touch. And I think this fits many previous findings suggesting that the attenuation relies on motor prediction. Here I just put only two, but there are many studies supporting this. So we know that attenuation is observed during active but not passive movement condition. However, the gating phenomenon is difficult to rely on motor prediction because the stimuli you receive during the movement, this externally generated, cannot be predicted based on your movement. So this phenomenon must rely more on sensory information. And I think this suggests that the two phenomena have different computational processes and different neural implementation that have to be studied further. And I would like to thank Henry Kerson. He was my PhD supervisor, and the funding agencies, but especially you for spending time here with us. Thank you very much.
Thank you so much. Thank you for this amazing presentation. And regarding questions, one of these is, do you find significant differences between PCS in the attenuation versus the attenuations plus gating conditions? The suppression seems very similar in both, and I wondered, how do you know the same gating is not also present in the attenuation condition? As you said, that gating can be observed on passive and active fingers during movement?
Yes, the gating is observed during both passive and active movement conditions. Now, I think the first question was Can I? Could I see the? Yes. Okay. Yes. Do you find do you find a significant difference between attenuation and attenuation and gating condition? Yes, we found an extra decrease in the attenuation and gating condition in the PSC that fits very well with the literature that I presented at the beginning. That gating refers to a difficulty in discriminating self-generated and external generated touches during movement, but it also makes them feel weaker. The suppression seems very similar in both. We found significant higher, like we found the suppression for the self-generated touch much greater. If this is what's meant here. How do you know the same gating is not also present in the attenuation condition? In the attenuation condition, so the gating concerns only the limb that moves. Okay, so if you present electrical stimuli on the left arm, but you move the right one, the stimuli on your contralateral to the movement limb don't change. You perceive them with the same magnitude. And this is a very big difference between the attenuation and the gating literature. In the attenuation literature, you move your right hand, let's say, to touch your left hand, and you will find attenuation in both limbs at the moment of contact. But the gating is really, there are many studies from the 90s showing that it really concerns the limb that moves. And the further you go from the muscles you move, the smaller the gating effect is.
Okay. Thank you so much. We can continue with the second one. There are also studies pointing the sharpened responses to expected self-generated stimulus, for example, as the results presented by Emily before. How can you explain the inconsistency between studies that either support the cancellation versus the sharpening accounts?
Yes, that's a very nice question. So first, I want to say that this, I suppose you refer to the study on touch of Emily that argues that attenuation and gating is the same phenomenon, and or they try to explain the attenuation findings of the previous study with the gating phenomenon. I think that the data I showed you today really differentiate between these two phenomena. Also, it's important to know that this suppression phenomena, it's something that it can be found in many animal studies. Okay, so it seems that it's a generalized phenomenon across the animal kingdom, so it's not something unique to humans. Also, there are previous studies that show that this attenuation is a predictive process, not a postdictive process. It doesn't depend on double touch on the finger, and you only need to predict the quantum of your finger using the motor command. Now, I'm also very interested in seeing if there are these sharpening or cancellation effects. So that's why we are trying to replicate these previous effects with touch. And we have pre-registered a study when we are controlling some additional factors, and hopefully, like, we are in the middle of data collection, but I hope like in 2 or 3 months we can discuss even more for that. So to see where do these cancellation or enhancement effects come from.
Okay.
And also it's something that I'm also thinking is, I don't know how easy it is to make a translation from the visual domain, in which we know that there are many attentional effects there, to the tactile domain. So I think we need a lot of experimental study for this.
Okay. Thank you so much. And there is another question. How did you choose the range of intensity levels for comparison and test the stimuli? What is the best method, to choose the range?
I used, to be honest with you, I relied on previous findings. So from a study from 2005, and on my study using these 2 alternative forced discrimination tasks, we're using the same range. So I don't think it would really change if you work with different ranges, but it would be interesting if you can test near threshold stimuli. I'm always working with supra-threshold stimuli, 2 newtons, but it would be also interesting in maybe working with, lower intensity, I think, as the Nadia's preprint suggests.
Okay. And the last one, can you explain the role of the attention mechanism having gating?
Okay, it also depends on what you mean by attention. I can tell you something about the this active inference computational account that argues that when you move, you are tuning the gains of your proprioceptive or somatosensory information, and this parameter gamma can represent this attentional tuning, if this is what you mean. So I think that such a computational approach that treats attention like this tuning of the gates could really explain the gating effects. And now, if you mean top-down attention, like because the stimuli are really externally generated during the gating, they are presenting at any time during the movement. I don't know how you could attend more the stimuli since you don't know when they are going to occur.
Yeah. Okay. I think that is all. Thank you so much.
Thank you very much. Thank you very much.
Collaboration.
See you later. Yes.
See you later. Okay. Well, our last speaker in the session 2 is Maryam Agumelai from Tehran. She is assistant professor in the Department of Audiology at Shahid Beheshti University of Medical Science. She has several publications in high-ranked journal, invited talk, travel award for presentation in renowned international conferences, and recently published a book chapter titled Clinical Measurement and Application of Cortical Auditory Evoked Responses. Her research interest focuses on brain mechanism underlying sound perception, acoustic regularity encoding, and speech processing. In the present symposium, she is going to present aging, inhibitory deficit, and auditory deviance detection. Maryam, whenever you want, thank you again for being here.
Maryam is not accepting the invitation right now. I think she's having some trouble. Let's wait a little bit for her.
Okay.
I knew that she was having some trouble because this platform is banned in Iran, and I know that she's connecting through some kind of VPN and stuff. So let's see if she can actually connect. 1 second.
Okay. Hi. Hello, Maryam.
You need to activate the microphone, Maryam. On the top of your face.
Yeah. Yes. But we cannot listen you.
Yeah, Maryam, here on the top, you can see that you have your microphone that's on the top of your face. Here, you can see that the screen of the microphone. Yeah, you have to click on that. Yes.
No.
No, not yet.
Yeah. Maybe there are some trouble with the Wi-Fi.
Okay, can you hear me now?
Now, yes. Perfect,
Right.
Maryam.
I'm sorry. I think the problem was with my connection. Well, okay. And can you see my slides?
Yes.
Okay. Hello, good afternoon. First of all, I'd like to thank the organizing committee for such a nice and inspiring meeting. And thank you actually for inviting me. I'm very honored to be part of that. I'm going to talk about a project we did some time ago in collaboration with the Brain Lab. And we actually worked on the effects of aging on auditory deviance detection, specifically on the lower stages of processing. As you probably know, the auditory system can extract acoustic regularities from the ever-changing auditory scene, and at the same time can spot novel or deviant stimuli which are not conforming those regularities. This function is very important for the auditory system because it actually it is the basis for formation of auditory objects, also for segregation of auditory objects, which are necessary for solving the cognitive party problem and also for understanding speech in difficult and noisy environments. And from the findings of recent studies, we actually know that deviance detection occurs at 2 stages. The higher stages at the level of mismatch negativity, with which we are all familiar. We know that mismatch negativity has been traditionally associated with change detection in the auditory system. But recent studies have shown that there is also a lower stage in the level of mid-latency responses. And the studies have shown that these 2 levels somehow function differently. For example, only simple deviations in basic acoustic features, such as frequency, location, and intensity, can be detected at the level of MLR by modulation of mid-latency responses. If we use complex ones, they can be at the level of MLR, and they can only be detected at the level of negativity. So we know that the auditory deviance system has levels of processing, and 2 levels somehow function differently. This is quite big. Well, the aging, that aging has some changes in neural and also in cognitive abilities that might adversely affect auditory detection. In fact, there are many studies that have addressed the issue, but previous studies have used only mismatch negativity. That's why we decided to look at the and to investigate the effects of aging on both mid-latency responses and mismatch negativity to see actually what's going on there. That we recorded both mid-latency responses and also the mismatch in a group of young and elderly during a frequency oddball paradigm. 29 normal hearing adults, older adults actually, and 33 young adults participated in our study. They all had normal hearing, and they were all right-handed, and we matched the groups based on gender and educational level. During the experiment, we asked the subjects to watch a silent movie and to ignore the auditory stimuli. Our experiment consisted of 2 conditions, an oddball and a reverse oddball condition. The oddball was actually a frequency oddball paradigm in which the 1200 Hertz tone burst was used as the standard, and the 800 Hertz was used as the deviant tone. During the reverse oddball paradigm, we somehow switched these frequencies in their roles as standard or deviant, so that we could be sure that we are comparing auditory voice potentials to the tones with similar frequencies. Actually, we did that to control for the physical properties of the stimuli. The probability of deviant stimuli was 20%, and we used an SOA of 300 milliseconds. In total, we had 1,200 stimuli for deviant and the same number of standard stimuli. So during the EEG recording, we actually recorded EEG from 32 electrodes, and we used 4 additional EOG electrodes for monitoring eye movements. All electrodes were referenced to an electrode on tip of the nose, and we digitized EEG using a sampling rate of 2048, and the data was filtered from 0.05 to 500 Hertz. For data analysis, we used different parameters for long latency and mid-latency responses. For analysis, the LLR, we re-referenced our data to the mass states to get a larger amplitude. And then we filtered the data from 0.1 to 30 Hertz, which is actually appropriate to get these like long latency responses. And we used ICA to remove the eye blinks and also the myogenic artifacts. And finally, we extracted epochs of 500 milliseconds and averaged them separately for deviant and standard stimuli. Voltage differences with more than minus plus 80 microvolts were rejected from analysis. And for mid-latency response, the process was the same except for the filter and duration of epochs and artifact rejections, which were different. What we got, if you look at the long latency range, we got a mismatch negativity and the following P3A response from the difference waveform between the standard and deviant in both young and elderly groups. And we actually did this statistic analysis using a mixed ANOVA with a within factor stimulus type and the between factor of age to look for the effects of age on these long latency responses. So what we got a significant effect of age on both mismatch negativity and the P3A. Actually, the amplitude of mismatch negativity was significantly reduced in the aged compared to the young group. And if you look at these plots, they show the distribution of our individual data for both mismatch negativity and the P3A, and in both young and elderly groups. This finding, I mean, the reduction of mismatch negativity amplitude has been found in previous studies as well. But in the mid-latency response, we obtained the MLR components, including PO and APA, MB, PB, and also NC in both groups for standard and deviant stimuli. And after doing statistical analysis, we found a main effect of stimulus on MB component in the young group, which means that in the young group, the deviant frequency was detected at the level of MLR on NB component. The amplitude of NB was significantly larger for deviant compared to the standard tone. But in the elderly, there was no significant difference between NB deviant and standard responses in the age group. Here again, you can see the distribution of our individual data. And if you look at the red plot, it shows it is for the elderlies. And this is actually the difference between deviance and standard response for MB component. And as you can see here, the mean average difference was quite close to 0. So if we look at both mid-latency responses and also mismatch negativity in both young and elderly group, you can see that in young group, we could see a clear mismatch negativity response, and also we could see the deviance-related changes of the mid-latency responses. But in the elderlies, the amplitude of the mismatch negativity significantly decreased, and there was no evidence of deviance detection at the level of MLR. Before I explain what does it mean in the aging and how aging affected our results, I'm gonna explain what we got in the young group. In one of our previous studies, we showed that in the lower stages of the auditory hierarchy, what is actually encoded is acoustic regularity, and it is encoded through adaptation of the response to a repetitive standard stimulus and a release from such adaptation with any change in the stimulus. But it seems that at higher levels, I mean, in the level of mismatch negativity, the system encodes for both acoustic regularities and also deviance detection. So back to the aging, if you look at the components of MLR in the elderlies, I forgot to tell, we also compared statistically the amplitude of different MLR components between the young and elderly, and as you can see here, they are quite enhanced, and there was a significant enhancement of NAPA, PB, and also NC amplitudes in the aged compared to the young group. The enhancement of auditory evoked potentials in the aged, I mean, as a consequence of aging has been reported before using both auditory and visual stimuli, and is actually in line with the hypothesis of a general inhibitory deficit in their auditory system. So it seems, I think the most striking finding of our study was lack of modulation of NB at the level of mid-latency responses in the aged group, which we think might be due to the insufficient inhibition of the, actually to the insufficient of the inhibitory mechanisms. With that, the response to the standard stimuli, which were repetitive, actually couldn't have been suppressed very effectively, and so the occurrence of deviance couldn't have resulted in release from such adaptation and to modulation of MB response. And if you look at the LLR response, I mean at the mismatch negativity, you can see kind of similar patterns of results. If you look at this blue line, which shows the response to standard stimuli in both groups, in the young group, we can see the effective suppression of the N1 in response to standard stimuli, but there is an enhancement of N1 to standard stimuli in the aged group, which has been actually reported in previous studies and is a result of, again, the inhibitory deficit in the central nervous system as a consequences of aging. So it seems that this N1 is a component of a slow wave, namely the repetition positivity, which attenuates with repetition of standard stimulus. And so inefficient suppression of this stimulus has actually resulted in a reduction in mismatch negativity. This again shows a deficit in acoustic regularity because this repetition positivity is actually a marker of acoustic regularity encoding, and also is a marker of formation of trace of memory. So this somehow again shows that there is a deficit in acoustic regularity encoding, and so we reported also a diminished P3A, which is quite normal because when deviant stimuli are not processed well, they would be, of course, less orienting. So in conclusion, it seems that aging mostly affects regularity encoding along the auditory hierarchy, and it begins very early in the auditory system from very early stages, and it affects the auditory deviance detection on both levels. Actually, it will result in the loss of deviance detection at lower stages, and it will diminish the effect at higher stages. And as an audiologist, I think at least some of difficulties that the aged population experience in understanding speech in difficult and noisy environment might be the result of a deficit in acoustic regularity encoding. Thank you for your attention. I want to thank my co-authors, Dr. Zahra Jafari, Katarzyna Żarnowicz, Dr. Sabine Grimm, and especially Professor Carlos Cicero from the Brain Lab, who helped me with this project. Okay, thank you.
Thank you so much, Maryam, for your participation. Although the troubles, at the end, we can have you as a guest, and thank you so much.
You're welcome.
There are one question, and it's Maryam. Thank you very much for your participation. You compared young versus elderly groups. What would you expect if you have tested infants?
Infants, of course, I mean, it depends, infants of which, I mean, at which age? I mean, do you mean newborns or the infants, for example, until 28 months? What do you mean by infants? I mean, can you define the time range? I mean, if we look at the infants, of course, the maturity will affect the results. Maybe we can see the, I don't know, but maybe I'm just assuming. I think maybe we can see the effects on the MLR as I mean the maturity effects, because the auditory system usually get matured from caudal to the rostral parts. So we might get, it depends on the stage of development that when we are looking at these responses. If we look at the responses of the newborns, we may not be able to get the mid-latency responses. I mean, because they are not matured yet. But if we look at later stages, of course, we would be able to get the mid-latency responses and also the device detection related responses as well.
Okay, thank you so much, Maryam. And there are comment regarding the paradigm used, and this speaker via this suggests that maybe using a cascade control, your results in the MMN could change. Do you think that could happen in your study if you change the paradigm for the cascade control?
Well, they might change, but actually this was a replication of a previous study which was done actually in the young group using the same paradigm, and they have used this cascade control paradigm. And so I think even if they change, I mean, there is something, there is a problem. I mean, even if the probability of the stimulus is affecting the response, I think they still cannot encode for those probabilities. I mean, there is still a problem because it's there, the effect is there on in young individual, but it's disappeared in the aging group.
Okay. That's all, and thank you so much for your participation again. And now we'll have 10 minutes for break, and then we move on the third session. We can go directly on the third session, clicking schedule on the top left of the website. Okay. Thank you, all of you, and we will see you again in the roundtable. Thank you.
Thank you.